So we're faced with the question, well, why, given that these stem cells are there and there's a clear need to repair, why does repair gradually fail as people with MS get older?
I'd argue that every trial that's been done so far, we've learnt something which has informed future trial design.
My vision is that in 15 years' time, if you're diagnosed with MS, you'd be given 2 or 3 pills, one anti-inflammatory, one pro-remyelination, and perhaps one other neuroprotective.
Hi, I'm Will Brown. I'm a neurologist in Cambridge, and I spend most of my time doing academic research into remyelination and big data.
Hello, my name is Alasdair Coles, and I'm an academic neurologist in Cambridge doing trials on remyelination.
And I'm Brett Drummond. You're listening to the ECTRIMS podcast, a new sounding board for the MS research community and the official podcast from the European Committee for Treatment and Research in Multiple Sclerosis. In today's episode, we're going to be chatting to Alasdair and Will about clinical trials for remyelination in MS. Myelin repair is considered by many to be the current holy grail for MS research. However, finding effective remyelination therapies represents one of the biggest challenges faced by MS researchers. Today, Alasdair and Will are going to discuss the concept of remyelination in MS and a promising clinical trial that they are currently involved in. Alasdair, Will, thanks for joining us today.
Thank you.
Thank you.
So remyelination is probably one of the most discussed topics in MS research by both researchers and patients. We know that myelin repair is possible, but for some reason stops working in MS. As a starting point, can you talk to us about where we're currently at with our understanding of myelin repair and the dysfunction in this process that occurs in MS, Alasdair?
Well, firstly, the great news about this conference is that we see remyelination way up the agenda. So I agree with you completely that this has become mainstream in a way that it hasn't been before. Mm-hmm. So in terms of the biology of remyelination, what we've learned over the last few years is that remyelination definitely happens naturally, particularly in younger people with MS. And there's even signs of remyelinated lesions in older people. So that capacity seems to be there. We know that the stem cells responsible for oligodendrocytes, sometimes called oligodendrocyte precursors or OPCs, are present in the human brain. Mm-hmm. So that was big news back in the 1990s and early 2000s. So we're faced with the question, well, why, given that these stem cells are there and there's a clear need to repair, why does repair gradually fail as people with MS get older? Mm-hmm. And we're beginning to hear the results of experiments which show why that's happening. So there are physical reasons and chemical reasons and biological reasons. Mm-hmm. So as we get older, our brains get stiffer physically, and that seems to be an impediment to repair. The debris that accumulates around demyelinated lesions itself, just the garbage of the pathology, gets in the way of repair. Mm-hmm. And then there are fundamental issues with the oligodendrocyte precursors themselves. Intrinsically, as they get older, means it's harder for them to differentiate and to repair.
Will, anything that you'd like to add to that? How do you feel like our understanding of this process has improved recently?
I think I agree with everything Alasdair said. I think the key bit to just reinforces the rationale here that, we have this slew of immunomodulatory treatments, and the unmet need is how to reduce disability, how to prevent it happening, how to reverse it. And that's just a really important thing to keep in the back of our minds. What we're trying to do is deal with the biggest unmet need in MS.
And so I guess considering this, there have been a number of attempts to identify compounds that may be effective at promoting myelin repair in MS. Can you talk us through, I guess, the history of that process, some of the trials that have happened, and what have been the difficulties that have really been faced to this point?
So the first step is trying to identify drugs or putative drugs that might have any impact on remyelination. And what's been exciting has been the varied ways in which basic scientists have sought to address that. Mm-hmm. And these are quite reductionist. So a nice example of that is the arrangement of many, many thousands of mini pillars, tiny little conical structures, with oligodendrocytes surrounding them, and then given a whole slew of drugs which could be repurposed to see whether in that very artificial situation the oligodendrocytes wrap around the pillars. So that's the very reductionist end. And then there's, as exemplified by Robin Franklin's work, animal-based in vivo work where demyelinating lesions are given to animals in the absence of inflammation, that's a key point, to see whether this or that drug given to the animals will promote repair. So there's a whole range of research leading to drugs being used in humans. And we now have several trials, which I would argue are all in their own different ways successful, in that we have multiple challenges. So not only is the issue of finding a drug that remyelinates the core issue, but how would we know if that's happened?
Yep.
How do we measure that? And how do we measure that at a sort of basic structural level or physiological level? And how does that relate ultimately to the experience of someone with MS? How long do these drugs need to be given? What's their effect once they've stopped, and so on? So these are all very big questions which we're just beginning to answer.
Yeah, definitely. And I guess to talk about some of those specific trials, bexarotene was a compound, and I think this links nicely to what Alasdair was just talking about in terms of thinking about a trial which may have been considered unsuccessful, but was actually quite successful in terms of allowing us to see how we might be able to monitor some of these things and seeing that it is actually possible. Can you talk about that a little bit?
Yeah, I completely agree. And I'd argue that every trial that's been done so far, we've learned something which has informed future trial design. So, I'll come on to the bexarotene trial, but even some of the smaller studies that, haven't, we don't discuss as much. One of the H3 antagonists, so there's a GSK molecule, this was published quite a long time ago now. Even that started to tease out, how do we detect that improvement? And for what it's worth, their method of doing it was to use lots and lots of scans in each patient, and that has, yes, it detected a small effect, but it doesn't, to me, seem a feasible approach for trial design. I don't know what you think, Alasdair. And then into obviously the Ari Green paper on clemastine, the first to find a positive results, and they were the first, or one of the first, to use VEPs and to use a crossover design. That seemed a very powerful thing to do and very informative. So moving on to bexarotene, recall that all of these trials were being planned around the same time when we just didn't know what to use. And so there we used MRI as our main outcome measure. And as you know, the outcome was negative. We used magnetization transfer ratio, or MTR, a very crude metric that does correlate with myelin. But in that, we did not see a treatment effect. But subsequent work, all pre-planned, has shown that actually if you look at a smaller level, instead of looking at a whole lesion, if you just look at each dot within the lesion, each voxel, you do see a treatment effect. And then that mirrors what we were seeing with visual evoked potentials. So again, what do we take from that? Well, RXR gamma agonism does seem to work, and we've learned how to do the trials better. And now the focus for an RXR, for that story, is to make the specific agonist that gets around the side effects.
Yeah, okay. Interesting. And I think there's an important point there, and I'm leading into a question, but this notion of learning from trials that are seen as unsuccessful, I think, is a really important thing to keep in mind, because certainly with the Biogen anti-lingo trial that you mentioned, it seemed to put quite a black cloud over the concept of remyelination for a while, having this trial not work. But as you say, actually we learned a lot from it, and we shouldn't necessarily think so negatively about it. In terms of designing trials around remyelination, how do you both feel we are in terms of getting close to understanding the best ways to do that? Obviously a work in progress, but are we getting closer to that?
So I think if you compare where we are with the history of designing trials for relapsing remitting MS with an anti-inflammatory treatment, we're back in the 1980s. That's where we are. So we don't know how long trials should be, we don't know how many people should be in them, we don't know what outcome measures to use for sure. Personally, I found that very exciting. I think it's much more fun doing this sort of work than it would be doing yet another study in relapsing remitting MS. And essentially, we're taking a gamble with any trial design. We're elevating one outcome measure as primary and putting others as secondary, but we don't really know. So that's fun for the researchers. I think it also demands some mature understanding of people who read these trials. It's very easy to dismiss a trial because a primary outcome measure is not met, but that's to lose all the useful information there is in the rest of it. So where are we with the design? I mean, if I designed a trial today, I would make visual evoked potentials the primary outcome measure, because that's the one that's given us the key hit with the Ari Green study and Bexarotene. That was a hit. But I wouldn't leave it there, because I think that on its own is too fragile an outcome measure in terms of our confidence in it. Instead, I would always add an MRI outcome measure, and there's a possibility in the future of fluid biomarkers. Because what I would look to as a sympathetic reader of these trials is convergence of multiple outcome measures on some sort of an effect.
Yep. Well,
yeah, nothing to add to that. It's that convergence that actually, to me, was the most convincing result in our trial. There's evidence from visual evoked potentials. There were signals in the MRI outcomes initially, and now we've got much harder evidence in MRI. So yeah, I agree. I think there should be more than one, and I fully agree. Even with my MRI sort of background, VEPs are the more sensible choice as the primary outcome measure.
Can I just mention one thing we haven't discussed but is really important is which patients should we try these drugs out on? So there's a classic discussion about whether it should be relapsing remitting patients or patients with progressive disease. I think I'd want to make a strong plea that we identify patients who have demyelinating lesions but intact axons, because clearly those are going to be the ones that are most easy to remyelinate. And here the rebuild trials, so Ari Green's trial, really sets the way forward there. Because if you use the optic tract, you can identify demyelination functionally with VEP, and you can identify that axons are pretty much intact by OCT. So that's a really nice way of looking at things. But more than that, there's a question of, well, do you want acute inflammatory lesion or do you want a chronic stable demyelinated lesion? And there are strong arguments either way there. I think some drugs may well work better in the acute inflammatory environment to promote remyelination, which of course is when naturally you would see remyelination. But people with MS would want a drug that remyelinates even the most chronic, stable demyelinated lesion. So even at the level of, who should we have in these trials, there is uncertainty.
I guess important that we are very mindful of that and at least stratifying who we're including to make sure that we're not getting negative results just purely based on cohort selection.
Completely. And that goes back to the early experience with immunotherapies, where we know now that we got things wrong because we chose the wrong patients.
Is there any suggestion at this point, or and this is almost an impossible question to answer, but I'll pose it anyway, that there will be cohorts of patients that we think just won't respond to a remyelination therapy? People with progressive MS, people later in their disease course, or do we think it may just require different drugs, that it may not be a one-size-fits-all?
I mean, I would argue someone who had few axons left is not going to respond that well, and those will be people with advanced disability and atrophy in the progressive phase of the disease.
I think I'd add to that, we're often accused, rightly, of concentrating on people with relapsing remitting MS. And I think from the person with MS's perspective, that can be quite difficult to swallow. I think if we can find a treatment that works in patients with relapsing remitting MS, then is the time to try it in those with progressive MS, for exactly the reasons Alasdair has outlined. Getting to your question on is there a way to pre-identify people who will do better? Well, there's some subtle ways, but there's growing evidence that some people have a remyelination profile, and that can be identified in various ways, but it's not ready for clinical practice.
So you're both currently involved in a trial investigating potential remyelination therapies. Can you tell us a bit about the background that led to the journey that led to the initiation of that trial?
Yeah, so I'll kick off there. So the big kind of scientific point here, which isn't ours, it's Robin Franklin's and colleagues, is that remyelination fails as you get older in animals and probably in humans. So the question is, how can you wake up the oligodendrocyte stem cells as you get older? And he's done experiments which first showed that alternate day fasting in rats led to rejuvenation of the oligodendrocyte stem cells. And by kind of analogy, thinking of the metabolic effects, the use of metformin would mimic alternate day fasting, and therefore metformin was used and proved to be successful in rejuvenating older animals, making their stem cells more liable to remyelinate. So we showed with bexarotene that any of our participants aged over roughly 40 did not respond to the drug. Okay. The younger you were, you did respond. So we think this age effect is real. And so we're using metformin in our trial to try and rejuvenate the oligodendrocyte stem cells. But Robin Franklin showed in animals that metformin on its own is not enough. You need some sort of trigger to get these stem cells mobilizing and differentiating. And we're using clemastine, the drug that Ari Green and colleagues first used, which experimentally works well when combined with metformin in older animals. So that's the scientific background.
Will, anything that you want to add on to that in terms of what sort of led to this?
Not so much led, more on the how are we measuring it. And I think this is, again, building on previous work, that we know multiple ways, or there's multiple MRI ways of detecting remyelination, and so we're excited to be measuring multiple ones on this trial to try and figure out for future trials what to do. That's about all.
So I guess just to ask a question there, that the initial studies looked at alternate day fasting. Is there a reason why that's not been included in this trial and you've gone with metformin?
So absolutely, I would like to do a trial of alternate day fasting. And indeed, there is a poster here at ECTRIMS about alternate day fasting in people with MS. But we found when we put it to a group of people with MS that considered this diet being required for 6 months, that they said essentially they wouldn't tolerate it. So we're taking that very seriously. We take the experience of patients with MS very seriously. And so that's why we're using metformin.
Fantastic. This idea of co-design with patients in mind, I think is really important and something we've heard a lot about at the conference as well. So can you talk to us then about the current status of the trial? Where are things at the moment?
It's exciting. So we're aiming to recruit 50 people, so half on placebo, half with metformin and clemastine. And at the moment, I think we've just hit 23. So our first participant has completed the trial as well, and so far things look well tolerated, and we have no readout on any effectiveness yet.
Okay. And I guess when can we expect then to hear any preliminary results?
Yeah, if all goes to plan, we'll be cracking the code at the end of next year, with first results in 2024. I should say that we do have an interim analysis planned, really, which is going back to a previous part of our discussion, which is just for people to check that our outcome measures are behaving as we anticipate they would, and that our power calculations are reasonable. So that may potentially shorten, but it may also lengthen the time this trial takes.
Yep. Now, obviously, recruitment for the trial has been made more difficult based on the global pandemic that we've lived through. Obviously, still trying to recruit for participants, for people that may be listening to this podcast episode. Do you have any message for them in terms of how they may be able to participate or send participants to you if they're healthcare professionals?
Yeah, sure. So this trial is very demanding for the participants, 7 long visits over 6 months. And we're essentially saying you need to be living quite close to the trial centre, which is Cambridge, UK, for this to be feasible. But for those people with relapsing remitting MS who are under the age of 50, living a couple of hours away from Cambridge, we're very open to seeing them and seeing if they're eligible.
Okay, fantastic. So obviously we look or wait with excitement to hear some answers and preliminary data from this trial. But to sort of put the overall topic into some perspective, a couple of provocative questions to finish off. Firstly, to both of you, do you think we will be successful in finding an effective remyelination therapy?
Absolutely, definitely.
Agreed.
Do you want to be more convincing with your answers?
Nope.
Okay, well then my second question then, considering the convincingness with which you both answered that, what do you think is a realistic timeline for this to occur?
So let me just justify myself a little bit.
Yep.
So if you were to ask the question, well, what has the most likely traction to keep axons healthy and alive in people with MS post-inflammation? And you say, well, what does the animal literature tell us about neuroprotective drugs, about remyelinating drugs and so on? There's no doubt that it's the remyelinating drugs that are most effective at being neuroprotective. Mm-hmm. So if we want to preserve axons, definitely the remyelination route is the one that the animal literature would support the most. And there are drugs that work in animals. Mm-hmm. So the reason why I'm confident is I think this is just a matter of translating what's happening in animals into people with MS. And that's not easy, but it is tractable.
Will, you were equally as confident in your answer. What do you think a realistic time frame is for this?
I think whatever I say, I'll be wrong. But I think if we take average trial durations, I would hope that there will be another positive phase 2 trial within the next 5 years. I hope there'll be multiple ones. Mm-hmm. Of course, the time frame from that to lead through phase 3 trials and then licensing. I think it's realistic to expect that in 10, 15 years, there may be drugs nearing market. Alice, I don't know what you think.
Yeah, I think that's fair. I mean, my vision is that in 15 years' time, if you're diagnosed with MS, you'd be given 2 or 3 pills. Mm-hmm. So, one anti-inflammatory, one pro-remyelination, and perhaps one other neuroprotective. I think that's a very reasonable expectation.
And do you think then if we have, just to finish off, we currently have really effective immunomodulatory drugs that can sort of slow damage of the disease, slow new damage. If we also have an effective remyelination therapy and we're learning a lot more about how lifestyle interventions can also help manage the disease, do you think we will be getting close to having almost complete management of MS for some individuals if we have all of those options available?
Yes, I thought for one horrible moment you were going to say cure there, Brent. So definitely not a cure, but I agree. I think we're moving a bit like our rheumatology colleagues are with rheumatoid arthritis to saying you will have this disease for the rest of your life, but we will contain it and it will not be a significant problem for you.
Yeah.
Agreed. I think it's really important to manage expectation. If we don't fully understand the full processes going on in MS, which we do not, then saying that we can treat every aspect of it is unfair. It's incorrect. So I see, like with Alasdair, having multiple therapies and even potentially multiple remyelination therapies. There are so many mechanisms. Most trials have concentrated on how to turn an oligodendrocyte precursor cell into something that makes myelin. There's multiple other mechanisms. Some presented at this conference that suggest that there are other ways of doing it too. So it may well be that even within one therapeutic strategy, there will be multiple drugs.
Yep. Well, it's absolutely a fascinating and an exciting area. We thank you, Alasdair and Will, for your time today to record this podcast.
Thank you.
Thank you.
And thanks everyone for listening. Once again, I'm Brett Drummond, host of this podcast episode and co-founder of MS Translate, a science communication initiative bridging the gap between people living with MS and MS research. You can read more about this topic and more on the ECTRIMS website, ectrims.eu. Do subscribe to the ECTRIMS podcast on Spotify and Apple, and follow ECTRIMS on Twitter, LinkedIn, Facebook, and YouTube.